We'll begin chapter 3 by looking at cellular structures and going through those first. What we have here is the mitochondria. The mitochondria is membrane bound, so what we have here outlined. All right, and then within that membrane, it has this inner cristae, and then it has of membrane folds, and then this outer membrane around the organelle itself. The cristae has many proteins on it for the electron transport chain and allows for lots of ATP generation, okay? And so the inner membrane then also projects into the matrix, which the matrix, here I'll highlight, is going to be this space where there isn't the cristae, okay? And that helps to increase then the surface area for ATP production, which will be produced by oxidative phosphorylation. To produce, one of the byproducts too is that the mitochondria produce reactive oxygen species, and this will eventually kill cells and happens as we continue to age. All cells have a mitochondria except for red blood cells or erythrocytes, and can vary from hundreds to several thousand within a different type of cell, depending on what the needs are for that cell. So areas that need a lot of ATP include like skeletal muscle and the brain as well. This next structure that we're looking at, this is the cytoskeleton, and these are going to be composed of microtubules, microfilaments, as well as intermediate filaments. So all of these straggly lines within the cytoplasm of a cell. These are to help organize the intracellular environment and allow for movement of cellular components, so like within this organelle, to move it from one side of the cell to the other. And so really, the purpose and function of the cytoskeleton is to act as a railway system to move stuff around the cell. These also are important for spindle apparatus formation to help pull chromosomes apart during mitosis. The next structure we're looking at here now are lysosomes. And so lysosomes are important because they're like our little Pac-Man of our cell, and they have digestive enzymes. So when they digest bacteria, like within this example, or some type of foreign material or waste product even, what happens is that it forms with a vacuole, and what it then helps to decrease and degrade that damaged organelle. And then so molecules can be destroyed and then will be shipped out of the cell. So proteins also can be digested, especially if they've been like misfolded or just are not properly in their right structure anymore. Also, the lysosomes are responsible for killing the cell via apoptosis, and this is where it releases its digestive contents into the cell, and so this is also termed as programmed cell death as well, and it happens naturally and continues to increase as we advance with age. So the big thing with this is that there will be fusion with some type of food vacuole after the cell has been ingested, and so this will create some type of primary lysosome where it has just the digestive enzymes, and then the secondary lysosome will contain part of the partially digested contents. So here, this would be an example of a primary lysosome, and then so here would be an example of a secondary lysosome. And then we have residual bodies that fill with waste and are expelled via exocytosis. So here would be an example of a residual body in terms of that lysosome. Next are peroxisomes, and so these are very similar in function to lysosomes, except two different features. First of all, it has enzymes for oxidative reactions, and so these are important for oxidizing toxic molecules. For example, formaldehydes and alcohols. So we have a lot of peroxisomes that are found within the liver. And so the ethanol then is oxidized to acetaldehyde, and so thus when we drink alcoholic beverages, it's not as toxic to our bodies because of that conversion. Also, there's enzymes that remove hydrogen and transfer to oxygen to make hydrogen peroxide. And so that hydrogen peroxide then has another enzyme called catalase that can convert to water and oxygen. These tend to be smaller in size compared to lysosomes and are very important for metabolism of amino acids and lipids, as well as production of bile acids. Here we have ribosomes, which ribosomes are going to be these little dots that we find. And there are two locations for ribosomes within the cell. We have them found here on the endoplasmic reticulum, but also they are free floating within the cytoplasm. These are made up of ribosomal RNA, so rRNA, hence the name, and of proteins. And so the function of ribosomes are to produce proteins based on genetic information that's contained from the mRNA. So then with the endoplasmic reticulum, these are going to be membrane passages from the nuclear membrane. And so here we have the nucleus with a nuclear membrane, and then here we'll have the different types of endoplasmic reticulum. And so there are two different types of endoplasmic reticulum. We have these that are closest to the nucleus, and you see these dark spots here. And so these are going to be the rough endoplasmic reticulum. And then we also have the continuation of these membrane folds, but that are not studded with ribosomes. And so those are the smooth endoplasmic reticulum. So within the RER, or here, so rough endoplasmic reticulum, what we have is protein synthesis because they're studded with ribosomes. Okay, and so we also have secretion, protein folding, or modifications of protein that occur within that rough endoplasmic reticulum. And again, this is closest now to the nucleus. Now the smooth endoplasmic reticulum, which is found up here, so SER, for smooth endoplasmic reticulum. This regulates and releases calcium ions, also helps to process toxins, and metabolizes fats and steroid hormones. Notice that it doesn't have much to do with proteins because there are no ribosomes that are on them. This organelle is the Golgi apparatus, and the Golgi are stacks of flattened hallowed sacs, and so these are also membrane bound. And so the unique thing about these are that they are C-shaped in structure as compared to the endoplasmic reticulum. So one side of the Golgi is going to face the endoplasmic reticulum. So one side here faces the ER and serves as a site of entry for vessels from the endoplasmic reticulum and that have cellular products. On the other side of the endoplasmic reticulum, it faces the plasma membrane. And so it takes transfer of these cellular products and then will expel them out of the cell via exocytosis so that it can go to other parts of the body. So the role of the Golgi apparatus then is to chemically modify, sort, and package different proteins, also transports lipids out of the cell, and so especially like with hormone production, and then creates lysosomes, which lysosomes will actually not be shipped out of the cell, rather they'll stay inside where they can then degrade different products. All right, and then lastly, we have the nucleus. The nucleus, here we have the nucleus, and then here we have the nuclear membrane. And most cells have only one nucleus. Muscle cells may have hundreds of them. Red blood cells, however, are the only ones that do not have a nucleus to them. Okay, and so that's why a lot of times red blood cells are, that term is used incorrectly because they actually cannot, or they do not have organelles. Big thing with the nucleus here, now this is what has that genetic information and contains our DNA. The nucleus has a nuclear envelope and it has two different membranes. We have an outer membrane here that's going to be continuous then with the rough endoplasmic reticulum, which here in the light green, this is what we see of the rough endoplasmic reticulum. Okay, and then also this inner membrane here that's going to surround that nuclei, and it fuses to the outer nucleus or membrane by nuclear pore complexes. And then, so what that allows for then are small molecules and RNA to move in and out of the nucleus through these different pores. And so these black dots here are the pores that we see. Now the plasma membrane of the cell, this is the has a phospholipid bilayer. If you remember, the phosphate heads of the phospholipid are going to be exposed to the outside of the cell as well as the inside. Can't see that. Inside of the cell as well as the outside. Okay, and again, that's because they are hydrophilic. They like water. The fatty acid chains or tails of those phospholipids are going to face one another, and then that creates that double membrane because they are lipophilic. They do not like to be in water. So the function of that plasma membrane then is to prevent loss of enzymes, nucleotides, and other water-soluble components of the cell. And so a big feature of that is this lipophilic membrane here. And so the hydrophobic center restricts movement of water and ions and anything that's water soluble. The hydrophobic or hydrophilic ends, or the polar ends here, then again have those phosphate heads to them. Now, for transport of materials, we also have these different protein channels, which tend to be very large in size. And these allow certain molecules to enter and exit the cell. For instance, when we go into the nervous system, this helps to allow sodium and potassium to either enter or exit the cell, respectively. The membrane also has proteins, like what we have here, glycoproteins, but also we have glycolipids as well. And these help to support, transport, and then have receptors on them, and are important for recognition of self. And so we'll get into that for the immune system as well. Another feature that we don't see on here though is cholesterol. Oh yes, we do. So here in the yellow, we have little cholesterol molecules found within the membrane. And so that's what is responsible with the fluid structure, dynamic structure to it. The cell membrane for us is not rigid and it's not stiff like it is for plants. And so that what that does though is allow for movement to certain areas. Like for instance, with our white blood cells, they're able to squeeze through different capillaries, like through the epithelium, and so to get to where they need to go. And so that's very important. Otherwise, we might have like blood clots that would occur. All right, next structure of the cell, and not all cells have this, but this is the cilia. And so here we see the wave-like projections that resemble a hair-like feature to them. And there are two different types of cilia that we have. The first is motile and the second are immotile cilia. So this example would be motile cilia, and so what they do is they beat in a row, like rowers in a boat, all in unison. And so we find this on the apical or the top surface of epithelial cells, and they line the hollow organs where they face each other. So we find this example within the respiratory tract to help move mucus and dirt and debris along and out of the bronchi, and then so it doesn't enter into the alveoli of the lung. And then also we find this within the female reproductive tract to help to move that ova along. The other type, the non-motile cilia, This is found actually on almost every cell and is important for like sensory function. So we find this like within the photoreceptors of the eye, and so to detect changes in light. And then we have different tubules, or found within tubules of the kidneys for movement of fluid. And then, so with the cilia, and we find with flagella as well, which are just going to be longer extensions of microtubules, is that they have the same arrangement. We have 9 pairs of microtubules arranged around a circumference of the cilia. So let's look at this in the next slide. So here, non-motile cilia will have these 9 sets of microtubules plus nothing in the middle here, okay? Difference with motile cilia then is that they actually have this, all right, within the center. So they have an additional 2 microtubules. All right, and so here's an example of flagella, and so the only cell within the body that has this is the sperm that has the flagella. And so these are going to also have the same arrangement of this 9 plus 2 of microtubules for movement. Centrioles are also going to be found within the base of each cilia, and these will be paired structures at right angles to one another and are involved for pulling chromosomes apart during mitosis. Microvilli are going to be shorter in size in comparison to cilia of both the motile and non-motile cilia, and these are found in special areas for rapid diffusion. So what the function of them are to increase surface area. So we find this especially within the digestive tract and within the kidney tubules. And they're important for, again, absorbing of nutrients for our digestive system, but then within the kidneys, it's for secretion and also for reabsorption of different materials so that so we're able to keep what we need in terms of ions and then also get rid of anything toxic rapidly. Now let's look into different ways that the cell can either take in or get rid of materials for each cell. So let's start with phagocytosis. And so phagocytosis is like cellular eating, all right? So this allows for bulk transport or large substances to enter into the cell. And what happens is that there's bonding with extracellular proteins to pull the cell forward by these pseudopods or these different feet. And so example of that would be like neutrophils and macrophages that do this. So let's look at this example where a pseudopod here, or a phagocyte, what it does is that it recognizes some type of bacteria, and so it's foreign to itself. And so it has these little feet that are going to engulf the bacteria and surround it, then it'll fuse together to form some type of food vacuole. Then what it will do is it will recruit lysosomes, and the lysosomes then are going to take that material and digest it. Other examples of other than bacteria that this would occur with would be dead cells will actually do this to themselves, or other cells will do this to dead cells, and then organic materials. And so this is very important for our immune system. This is important for the inflammation response and then also for apoptosis or cell death. Endocytosis now, this is where we're taking stuff in and bringing large amounts into the cell. And so the plasma membrane has these furrows that are going to project inward, okay? So this is going to be here, we have the outside of the cell, and then this is the inside of the cell, all right? And see, the plasma membrane is starting to invaginate this material on the outside of the cell. So a small part of that membrane then is going to pinch off, and it's going to form a vessel that is going to enter into that cell. And so there's two different types of endocytosis. The first one is pinocytosis. Let me write that down. All right, so pinocytosis, this is nonspecific. So anything that is in bulk, so a lot of times like different types of ions, molecules that the cell needs, it's going to take it up and bring it back in. The second type though is going to be specific, and so this is receptor mediated endocytosis. And so here now within the plasma membrane, say we have different types of receptors or different types of proteins that are going to bind to a substance to allow it to come in. And so we'll see this a lot with like cholesterol and also with neurotransmitters too that will bind to some type of receptor to bring it back in. Unfortunately, the AIDS virus and hepatitis virus are also receptor mediated, where they change that protein to recognize it as self, and so actually will bring that virus into the cell and then wreak havoc for the cells. All right, and then the last type that we have is exocytosis. So now it's the opposite of endocytosis, where we are removing products out of the cell. Okay, so here we are inside the cell in this picture, and then now here this purple is going to be outside of the cell. And so the Golgi apparatus packages proteins in this vesicle, and it eventually will start fusing with that plasma membrane, where then it will release its contents out into outside of the cell. So this is how we have transmission of neurotransmitters from the inside of the cell and to release it outside of the cell.